AlphaGenome, the latest release from Google DeepMind, is an advanced AI model designed to analyze 1 million DNA bases at a time. It doesn't just focus on individual genes. It maps the effects of genetic mutations across the full regulatory genome. Think of DNA as a programming language, and you as the software it runs. The human genome contains over 3 billion letters, yet less than 2% of that actually codes for proteins. The remaining 98%, it regulates things like gene expression, RNA splicing, and the 3D folding of DNA. But for decades, we've struggled to understand how this non-coding region really works. Now, that's beginning to change. AlphaGenome is a major breakthrough. For the first time, we have a single model that unifies long-range context, base-level precision, and state-of-the-art performance across a wide spectrum of genomic tasks. For years, genomic models faced a tough trade-off. They could either look far across the genome, but only see a blurry picture, or they could zoom in with sharp detail, but only on a tiny section. AlphaGenome changes that. It processes 1 million bases of DNA at a time, while still capturing changes at single base pair resolution. For the first time, we get both long-range context and pinpoint accuracy in one unified model. You give AlphaGenome a DNA sequence, and it tells you how that code functions inside the cell. It can predict gene expression across different tissues, where splicing happens and how it's used, which regions of the genome are open and accessible, like ATAC or DNA signals. It identifies histone modifications, transcription factor binding sites, even 3D contact maps that show how DNA folds and interacts with itself, all from just the raw sequence. No experiments, no lab work, just AI reading the code of life. AlphaGenome sets a new benchmark in the field. It outperforms specialist models on 22 out of 24 track-level tasks. It leads in 24 out of 26 variant prediction benchmarks. And it runs faster, using only half the compute required by models like Enformer. But what truly sets it apart? It does all of this in a single pass. No stacking models, no stitching through the exchange. Just one unified system, end to end. This isn't just theoretical. Researchers put AlphaGenome to the test on TL1 mutations linked to T1 leukemia. It predicted the emergence of a new MYB binding motif, activation of nearby enhancers, increased H3K27AC levels, and upregulation of the TL1 gene itself. A single base change, triggering an entire regulatory cascade. This is the first model of its kind. It understands RNA splicing on every level, from individual splice sites and junctions to full isoforms. It detects regulatory interactions across massive regions of the genome, up to 1 million bases at a time. It predicts how non-coding variants, the parts of DNA that don't code for proteins, still impact biological function. And most importantly, it can trace a change from cause to consequence. All of it down to a single nucleotide. AlphaGenome is changing how we work with the genome. It helps researchers identify the root causes of rare diseases and simulate mutations before any lab testing begins. It can be used to design synthetic DNA, precisely tuned for tissue-specific control. It also helps prioritize drug targets from GWS data and predict off-target effects in gene regulation, crucial for safe, effective therapies. All of this powered by one model. AlphaGenome is becoming the new backend for genomics. Like any model, AlphaGenome has its limits. It still struggles with very long-range interactions, those beyond 100,000 base pairs. It isn't yet optimized for individual, personal genomes. It predicts molecular effects, not full physical traits or disease outcomes. And it's not ready for clinical use just yet. But remember, this is only version 1, trained in just 4 hours, using half the compute of Enformer. The foundation is here, and it's only getting stronger. The real breakthrough with AlphaGenome isn't just accuracy, it's unification. Where researchers once needed over 10 models to study a single mutation, now one model does it all with a single API call. It's general purpose and ready to be fine-tuned for any context. DeepMind is already offering API access with a full release coming soon. First, AlphaFold cracked protein folding. Now AlphaGenome is making gene regulation fully computable. Every failed treatment, every rare disorder, every complex trait we couldn't explain, they all trace back to DNA we couldn't fully understand. But now with AlphaGenome, the system comes into focus. We're no longer guessing, we're seeing. And once you can see it clearly, you can start to shape it. Biology stops being a mystery and starts becoming programmable. This isn't just analysis, it's control. AlphaGenome is the turning point. Every disease has a signature. Every mutation holds a question. AlphaGenome is how we finally read the answers. The genome decoded.